right, BIM 100 students, the final is just around the corner, so let's do a super high-yield review of the key stuff from Lecture 8. We're going to focus on how you connect experimental data to the biology of cancer. My goal here is that by the end of this, you'll see these questions on the exam and know exactly what to do. Let's get right into it. So we're going to build this whole review around a classic problem. You might even recognize it from Homework 8. Picture this. You've got a virus, and it might cause cancer. We know it's messing with the cell's genes, but which ones and how? This is exactly the kind of puzzle Professor Pasquinelli wants you to be able to solve. To even begin to answer that question, we need the right tool. And for this job, our go-to is going to be the DNA microarray. It's honestly one of the most powerful ways we have to get a big picture view of what all the genes are doing at once. So what is a microarray? The best way to think about it is like a snapshot, or maybe a report card for all the cell's genes. Instead of checking them one by one by one, it tells us what thousands of them are doing at the exact same time. It shows us which genes are working overtime, which ones have been shut down, and which are just humming along like normal. Okay, so how does this magic actually work? Well, first you grab RNA from your two samples. In our case, that's our normal healthy cells and our virus-infected cells. Then, and this is the key part, you turn that RNA into something more stable called cDNA, and you tag them with different colors. Let's say green for the normal cells and red for the infected ones. You mix all that colorful cDNA together and just wash it right over the microarray chip. Now the cDNA from each sample will stick, or what we call hybridize, only to its matching gene on the chip. Finally, a scanner comes in and reads the colors. Simple as that. Now, reading the results is probably the most important part. If you see a bright red spot, that means a ton of red cDNA from the infected cells stuck there. So that gene is being cranked way up. It's upregulated by the virus. If you see a green spot, it's the total opposite. The gene is more active in normal cells, which means the virus is shutting it down. We call that downregulation. And what if you see yellow? Well, what happens when you mix red and green light? You get yellow. It just means the gene is expressed about the same in both cells. So just remember, red is up, green is down. That's the main thing. Okay, so now we know how the technique works and how to read the colors. Let's actually apply this and decode the data from our homework problem. So here it is, the simplified results. The experiment showed that genes A, D, E, and F were all upregulated. These would be our bright red spots on the microarray. But gene C was downregulated, so it would show up as a green spot. So we have the data. But what does it actually mean? What's the biology behind these color changes? To make sense of it, we need to bring in two of the biggest players from our cancer lectures, oncogenes and tumor suppressors. These are the two main types of genes that, when they get messed up, can push a cell down the path to becoming cancerous. So first up, we have proto-oncogenes. In a perfectly healthy cell, these genes are basically the gas pedal. Their job is to tell the cell, hey, time to grow and divide. They're absolutely essential for normal life. And that proto part is super important. It means this is the normal, good guy version of the gene. And you guessed it, if you have a gas pedal, you've got to have brakes. That's where tumor suppressors come in. Their whole job is to tell the cell to slow down, stop dividing, or even self-destruct if something goes wrong. They stop things from getting out of control. For a cell to work right, you need both the gas and the brakes functioning perfectly. Okay, this is where it all clicks together. Let's connect the dots. Our data told us the virus upregulated a bunch of genes, A, D, E, and F. Think about it. What happens if you slam your foot on a car's gas pedal and it gets stuck down? You get uncontrolled acceleration, right? It's the same thing here. Overactivating a gas pedal gene leads to uncontrolled cell growth. So those genes are almost certainly proto-oncogenes. Now, what about gene C? The virus downregulated it. What happens if you cut the brake lines on a car? Again, uncontrolled acceleration. By disabling the brakes gene, you get uncontrolled growth. So gene C must be a tumor suppressor. See how the logic works? Now, this whole exercise isn't just for homework. This exact way of thinking is what you're going to need on the final exam. So let's take a look at how this stuff actually shows up on a real test. This is exactly what Professor Pasquinelli wants to see. Can you look at some raw data from an experiment and draw a solid biological conclusion? It's always a two-step process. First, figure out what the data is telling you, and then apply the right biological idea to it. And look at this. This is not a hypothetical. This is straight from a previous final exam. It gives you microarray data from tumor cells and asks you what genes you might target for therapy. You have to look at the data for genes A through F, figure out which ones are up, which ones are down, and then label them as either proto-oncogenes or tumor suppressors. This proves that what we just walked through is 100% something you'll need to know. So here it is, your big pro tip for the final. It's a two-step dance. First, you state what the data shows. Is the gene upregulated or downregulated? Simple. Then, you connect that fact to its function. An upregulated gene that helps cancer grow is a proto-oncogene. A downregulated gene that helps cancer grow is a tumor suppressor. Always, always connect the data to the function, and you can't go wrong. All right, let's wrap this all up with a quick little checklist, just to make sure these ideas are totally locked in your brain for the exam. So here's the recap. Number 1, microarrays give us that big picture snapshot of gene expression. 2, the color code is your friend. Red means up, green means down. 3, connect the data to the function. Those upregulated gas pedals are your proto-oncogenes. And 4, those downregulated breaks are your tumor suppressors. You absolutely should be ready to look at a table of data and make these kinds of connections. And I'll leave you with one final question to chew on. Microarrays are awesome for seeing everything at once, but they aren't our only tool. If you wanted to double-check and confirm that, say, gene A really is upregulated by this virus, what's another technique we've learned in this class that you could use to prove it? Think about that, keep connecting the concepts, and you are going to be in fantastic shape for the final. Good luck with your studying.